Today, I'm going to talk about the power and promise of functional proteomics. So before I start, let me tell you where we are located. So this is our Purdue campus, and this is the Discovery Park, and this building here is the Binley Bioscience Center. So in the center, this building, the Purdue Proteomics Facility is located. So today, I have two missions here today. So in the first part of my presentation, I'll be talking about proteomics and definitely why proteomics. Everybody knows, but my goal here is that proteomics is not merely a technique where you generate the list of the protein. It's more about the biology. So I'll try to convince you and motivate you why this technology is very important in the current era of systems biology. And then I will be talking about different approaches, quantitative approaches we use to quantify the protein and characterize them by mapping their different modification and their interaction. And after that, so I'll try to highlight some of the research I'm doing here at Purdue in my independent research using those different tools, and also the future direction where I'm going myself for my own research, as well as the field, where the field will be moving in the future. So with this, everybody knows this, the information workflow here from DNA to RNA to the phenotypes. So the DNA, they have this protein code, the gene information to code coding information for gene, which needs to be transcribed into protein and then into RNA and then translated into protein. But the point here I want to make is that when you move from the DNA down here, so the more and more the proteins are more closely linked to the phenotypes. So the protein, that's why the information about protein can help us to link the different phenotypes we observe. And also there is a complexity there because proteins can be regulated by so many different types of post-translational modification and their network or protein-protein interaction. So then why we need a proteomics, though we have a very nice, very well-developed tools for RNA-based analysis. The point is, this is a nice paper published in 2010 in Molecular Systems Biology, which shows that the RNA can only give the half of the story. Only 27% of the phenotypes can be predicted or described based on the information we got from the transcriptome analysis, whereas the 40% of the information can be linked to the proteome analysis. So definitely these two technologies, they are very complementary to each other, and the proteomics can contribute a lot when we are talking about the biology, diseases, and the phenotypes. So that clearly shows that we need a proteomics tool to better understand the system's biology. So another thing is, if you look at the cell, so this graph here shows the copy number of the mRNA transcript versus protein. If you look at cell per gene, there might be in average 3 transcripts copies, mRNA copies per cell for a particular gene. But if you look at protein, that can go up to 1,000-folds more. And then there is another, the research where they show that there is either very poor correlation between mRNA expression and protein expression, just shown in this graph here. So this shows all 3 genes and their expression related to the protein expression. And you can clearly see these dots are scattered all over. And also that relation also depends, is very gene or protein specific. For example, if we take this mRNA1, it looks like there is some relation between protein and RNA, mRNA expression. Whereas if you go to the mRNA3 and protein 3, it looks like there is almost no correlation there. That also says that only the mRNA level the based analysis could not provide the whole picture what's happening into the cell. And the proteins, on the other hand, compared to the mRNAs, they're very dynamic, and also they are very they have a very large dynamic range, which I will come back later in my slides. So as I mentioned before, the protein is directly connected to the phenotype. So therefore, if we want to characterize the phenotypes, we need protein or proteome level information. So one good example here, you can see the 60 to 80% of the height, the genes that determine the height, there are 60 to 80% are controlled by genetically, and only 20 and 40% comes by nutritional changes. So there is a very famous paper recently published where the gene that's HMGA2, which is the height controlling gene, and the replacement or mutation of cytosine into thymine looks like it affects the height. In every time when there is one mutation, one copy of that gene coming from one of your parents, it looks like it can change the half centimeter of the height. If you get the both from your parents, it's going to change 1 centimeter. So it definitely, there is not only one gene, but the cluster of gene, or possibly hundreds of genes that are involved in determining this type of phenotype. So this emphasizes that information about the protein and proteome is definitely going to be critical when you are characterizing the different phenotypes. So the same thing, if we look, we can see the dynamics of the proteome. So we have about the 6 to 20 million ribosomes in a cell, which are producing at the rate of average 5 amino acid per second to synthesize a protein. So if you look at the total number of cells here, we have almost 40 trillion cells in human bodies. So, and of those, a significant synthesis going on. There is also the significant the degradation. 3 to 5% of the proteome is destroyed each day. So it appears that we are a new person in every 3 to 4 weeks. And the protein lifetime also ranges very differently. There it can go from 10 minutes to 72 hours. So that shows how dynamic the proteins are into the cell. But the one important thing in addition to that is the proteins are they function in a very highly concentrated form. So for example, the cell can have up to 300 milligrams of protein per milliliter. So if you compare that to the typical concentration you use in your laboratory reaction, biochemical reaction, it's a 2 to 3 order of magnitude higher. So that tells the proteins they're functioning in a very thick soup of this fluid into the cell. And when we design the experiment, these are the things we should always keep in mind. Because when we extract the protein using different buffers, we are diluting them substantially. So when you dilute, you are also affecting their characteristics, like their interaction, their modification status, as well as their functions too. And the one additional point I would like to make is there are so many different potential modifications the cell can have. It is predicted over 300 modifications can have in proteins in a cell. So that makes the proteomic analysis very challenging. But with that challenge comes a lot of novel information too, if we can design the experiment in the right way. So this is this slide where it shows the dynamic range of protein in human plasma. You might have seen this slide before too. And for example, if we compare the serum albumin here, which is the most abundant protein in our blood, and this is the interleukin. So these two proteins are, so if you compare this 7 or 8 order of magnitude difference in their abundances. Now, when we try to analyze the protein sample using this mass spectrometry, so we are trying to identify a very tiny feature there with this a very large feature, which can definitely suppress the signal coming from that tiny the target, which appear to be most of the biomarkers, interesting biomarkers. So we need to think all these larger dynamic range, the complexity, concentration, and how we can design the experiment upstream before we move them into the mass spec for their identification. So, and then I also compare, when I think about the cell, I also compare it with the school of the fish. Yes, the protein, they're not only static and they're not functioning in isolation. So they function as a community molecule, and they're very dynamic. They're migrating from one compartment to the other, and within the compartment from one location to the other location. So, and this movement and their function is strictly guided by certain rules and regulation, just like the school of fish, and they're moving in a certain direction. If a fish loses that direction, it will never be able to go back and join that path. Yes. And I often think about this, that we all know that this tragic accident happened a few years before, where the Malaysian airline was lost in the Indian Ocean. So why do that plane was never found? Because it's a small plane compared to the whole ocean there. When it lost the signal, there is no guidance to find it. So the protein also, when they lose that particular guidance or movement, then it's very difficult to find them their other components or their interacting partner to execute the function. So these all are the critical points to think before we design our experiment to test our hypothesis. So then the protein, as I mentioned, they're the community molecule. So then one thing they do is they communicate. They also help the cell to communicate across the different compartments. For example, this slide here shows this melatonin 1 receptor. When it gets activated, and it binds with this RGS2 regulator of this G signaling protein 20, and then once this binds, it activates, and this G protein, protein can go and can interact with the RGS20, and that helps to flow the information between these two compartments. So the protein, not only they do their function, but they also help to really to deliver that information, that functional information, from one compartment to the next compartment. And the other, when they carry out this enzymatic reactions, the protein needs to have a certain the structure. For example, here I'm showing you the glutamine synthetase from maize, which is the homodecamer. So when they form this homodecamer, then they can function and then convert this glutamate into glutamine. So the structure, the way the protein they structure, they fold, is also going to be critical when it comes to their functions. And another good example is the substrate channeling. What I'm showing here is this malate dehydrogenase and citrate synthase and this ecotinase. These 3 proteins in the TCA cycle, they have to interact with each other if they want to flow the substrates from one place, one location to the other location, or from one protein to the other protein. So that shows that the protein, they're not individual entities, rather they're very complex molecule working in coordination with other partners in the compartment. So this all information, if we design the experiment, so what I'm trying to emphasize here that so if we design the experiment in the right way, we have a chance to identify all those information using proteomics-based approaches, and which I will try to highlight in my the other slides in this presentation. So the proteomics, as I said, the most critical part when you design a proteomic experiment is upstream, the sample preparation. That guides whether our experiment is going to be successful or not. So broadly, the overflow can be divided into 3 parts: sample preparation, data analysis or data collection by using LC-MS or MSMS, and then downstream pipeline to glean that information and connect that information to test your hypothesis or generate new hypothesis or discover new biology. And you all know the proteomics is mass spec based proteomics. And you can realize it's a very complex sample. So when you extract the protein, and you're extracting thousands of protein, and typically we use this peptide-based mapping or analysis. That means we digest those protein into peptides using different proteases like trypsin, lysine, chymotrypsin, and so on. So from that thousands of protein, now we are generating hundreds of thousands of peptides, and these peptides are being analyzed by the mass spectrometer. So the mass spec cannot handle all these ions, all the peptides at once. So then what we need to do is we need to separate them. So we need to separate them by certain systems like the chromatographic system at the front of the mass spec. So that rather than putting all these ions at the same time into the mass spec, we are putting a certain number of ions as a function of time under certain time points. So we use this LC system. That's why it comes the LC-MS or LC-MSMS, and where we can separate them by the reverse phase, or by using the capillary electrophoresis, you can separate based on the charge. So you reduce the complexity of these ions when they're entering into the mass spec. So So these are the most, there's almost all the proteomic based the approaches currently use mass spec based approaches. These are few instruments I'm showing here that we have in the Purdue Proteomics Facility. This is a Q-Exactive, this Orbitrap thermo instrument. This was installed in 2016, and it's a very stable and a very powerful instrument to do large scale proteomics. And then we have the Orbitrap Fusion Lumos instrument, which comes with the electron transport dissociation, that ETD. It comes with the multiple fragmentation technology, and one is the ETD, which stands for the electron transport dissociation. It's more like the soft ion, the fragmentation technique, which is known to be better when we're doing experiment for post-translational modification like the glycosylation or phosphorylation. And then this instrument is more, is Endora, is a triple quad instrument. And if we want to do more targeted proteomics for absolute quantitation, this instrument is used and is available for you guys if you are interested to do the more absolute quantitation using this more targeted approach. And there is another 5600 triple TOF instrument we have. This is a little bit underutilized instrument, but it's also very powerful. And it can do both the targeted as well as non-targeted or global profiling. So this shows the typical the chromatograms and the information we can obtain when we run the sample into the mass spec. So I just mentioned before, we use the liquid chromatography, different chromatographic system up front to separate peptides before they enter into the mass spec. So here, what I'm showing in this panel, they say the TIC, which stands for the total ion current. And this line x-axis here shows the duration of LC separation. Depending on the sample complexity, you can use a different LC separation time. So let's say you have a very complex sample, then you may want to run longer gradient so that you are very slowly releasing those peptides before it enters to the mass spec. If you're only analyzing a less complex sample or only gel bands or one or two protein, then you might not need this long separation. So you can squeeze and make a shorter gradient to run into the mass spec. So definitely this LC, the separation makes a huge impact in the quality of the data and the depth of the proteome you can capture. So when you do this black, the line you see here, these are all the information you are collecting about peptides. So if you take this area here, now you can see this fully scan. What this is showing is these are all the peptide masses and the charge. So you know the mass spec identifies or separates those ions based on their size, the mass and the charge. So we can see multiple peaks in this fully scan, and what the software does, depending on what type of method the instrument does, depending on what type of method you are using, just like there is the method called the data dependent acquisition, and there is another is data independent acquisition. In the data dependent, you select, okay, in that fully scan, I want to fragment certain number of ions only. I'm not going to fragment everything because that may be too much for the mass spec to fragment everything you are putting there. So we do the data dependent. Then it picks those ions and then it fragments. It got all the fragment ion information there. And you know, in the nature, there are some isotopes, yes? So we have this carbon 12, carbon 13, and carbon 14. And so do we have this nitrogen 15 and nitrogen 16. As mass spec is very, very high mass accuracy measurement, we can also, for this peak, this is the peak coming from a peptide. So we can also measure their isotopic distribution. So this is called, everything is C12 and N14 here, but this is maybe at least one of the amino acid has either C13 or N15. So we can get this isotopic envelope, isotopic profile of his ion. And we use all this information, and also the peak height shows the intensity, signal intensity of that ion. So we took all this signal intensity, fragmentation information, and the time these ions are coming up from your LC system, as well as these isotopic patterns to identify, to sequence the peptide, and then the peptides later on map to the protein sequences. So you might have heard the sensitivity, speed, and resolution of the mass spec. This graph here shows one example how having a high resolution instrument helps us to accurately identify the ions. For example, if you run your sample in a 35,000 resolution, then there is only one peak. But if you can run an instrument in the 70,000 resolution or 140,000 resolution, you will see there are two peaks in 492.2495, and there is another peak here. You can nicely resolve in these two peaks. Now, here, there is only one identification. Here, you can identify two features there. So that's the advantage of having these high resolution instruments for proteomic analysis and any kind of analysis. And so the proteomics is not only identification, it's a quantitative tool too. The mass spec inherently does not have the quantitative power. So what we do is, before we put the sample into the mass spec, we design these different quantitative approaches so that we can glean the abundances of protein we are measuring. So this quantitative approach can be broadly divided into three categories. One, we do the label-free quantitation. What does it mean? That you don't have to put any label, anything in your sample. That's a label-free, as the name goes. And we do labeling. So where the labeling can be chemical labeling as well as the metabolic labeling. And I'll explain them in next few slides. And when we move this label-free approach to the labeling approach, the accuracy grows in this direction. That means the metabolic labeling can give much higher accuracy in our quantitative measurement. But there are some other disadvantages of each method compared to the others. So let's talk about this label-free approach. So in the label-free, you are running each sample independently. So that in that situation, one critical part is the retention time. So the retention time is the time where these peptides are coming off from your LC system before they enter into the mass spec. I mentioned before this 120-minute separation in our LC method. So what are these particular ions are exactly eluting at the same time? That defines how accurate your quantitation is. For example, this ion of interest, you can clearly see that in these three samples, they're very nicely eluting, very reproducibly eluting in a certain time point. But if you look here, there is a shift, like this sample has, the elution time is shifted in these two samples compared to the first one. So then when you go and align those peaks, to identify that ion, so you might likely lose, or the alignment might fail, which ultimately hurts your quantitation. So it is critical to have a very good chromatographic system, highly reproducible chromatographic system to accurately quantify your protein based on the label-free quantitation. So then there is another method which called the Silac method. We say this is the stable isotope labeling in amino acid in cell culture. So in that Silac, we have the heavy version where the carbon and nitrogen are labeled by C13 or N15. And then another one is the light, which is the normal one, C12 and N14. So the advantage of this metabolic labeling is now you can grow the cells with this light version of the amino acid and with the heavy one. And after you reach a certain level of concentration of a heavy, let's say 95% and above, then you can mix them together. So that now, instead of analyzing these two independently, just like I mentioned before for the label-free quantitation, now you can analyze them together. The only difference here is the mass, either the C13 and N15 mass, but other features, other characteristics of these ions are exactly the same. So what happens is now both of these ions, now you can monitor within the same elution time. So then they nicely, they overlap to each other. Then you can quantify these ions easily between these two samples. Now it allows you to accurately quantify, plus it also helps you to multiplex. Now with the Silac, you can at least do 3 samples at the same time. There is a heavy one, there is a light, there is a medium label, and the heavy label sample. So 3 samples can be mixed together and analyzed. But the disadvantage is because you are labeling with heavy, so you need to have a saturated level of this incorporation of this heavy label. Yes, I mean, if there is 95%, then still you have 5% of those ions are coming from the light version. So that might sometime interfere when you are analyzing, and it might limit some of your quantitation. The other thing is when you mix and multiplex them together, rather than measuring these two samples separately, now you are mixing. By mixing, you are increasing the complexity too. So that you should also consider, if you increase the complexity, should you go and fractionate those before you put into the mass spec or not? Or should you run more longer gradient so that you can identify as many ions as possible? So the third one is the chemical labeling. So in the chemical labeling, so the difference here is we are growing cells in vivo. We are growing cells in vivo with this heavy and light medium, but in the chemical labeling, which we call this I-tag, so this is isobaric tag for the relative and absolute quantitation, or there is another one we call the tandem mass tag, TMT labeling. So these are the labeling we do after we extract the protein and digest them into peptide. So these the tag here, they come with a different the reporter ion and the mass. There is a different mass here, which can be changed depending on how many the reagents we want to they use. So it can go, the masses can go, can range from 31 to 41. Now we have a duplex, quadruplex, and a 6, 8, 12, even 16-plex those reagents now. What does that mean is you can take those reagents and then you can treat each of your peptide sample, and then they go and they covalently label this the amine terminus, and then you can mix them together after you do the reaction. So each peptide, sample has one reporter ion there. So now if you have the 16-plex, that's 16 different reagents, you are labeling independent separately, and then you can mix them together before you run into the mass spec. So like, for example, if you are using the 6-plex, so you have these peptides labeled by those 6 different reagents here, and you can mix them together and run into the mass spec. So once you run into the mass spec, in the fragmentation process, these reporter ions, they get detached from your peptide. When you're fragmenting the peptide, you are releasing those reporter ions, and you can monitor the intensity of these reporter ions and compare them in your sample. So higher the intensity of reporter ion, for example, indicated by red here, indicates that the peptide in that sample, for that particular peptide in that sample, has more abundant than the rest of this peptide in the rest of the samples. So this allows you to analyze large number of samples at the same time. As I mentioned, you can go up to 16-plex sample there. But again, you have to remember there are other caveats as well. For example, is our labeling efficiency is acceptable? Are we labeling 100% of our peptides? That's the one thing. The other one, do we have, when you mix, you're increasing the complexity. And with that complexity also comes the challenges when you are separating in your LC system, just like I mentioned before about the metabolic labeling. So there is usually when we mix together, then we go and fractionate them so that we are, and one sample, this mixed sample can be fractionated at the peptide level into multiple of fractions before we put them into the mass spec. So we need to consider those other steps if we are going to use this chemical labeling approach as well. So far I talked the quantitation. So I thought this slide should be a later one, but let me explain. The proteomics is not only identification and quantification and post-translational modification. It also allows you to measure the protein complexes and protein-protein interaction. This is what I'm trying to show here is before we put the sample into the mass spec, we can use a different sample preparation technique. So one I'm showing here, this size-based gel filtration chromatography or size exclusion chromatography. What we can do is when you isolate the protein in the native form, endogenous form, then we can pass them through these the SAC columns and separate them based on their size. So once you separate based on their size, and then you can process them through the mass spec, and you can use this elution profiling. So for example, this is your SEC fraction, and these are the peaks there of this a particular protein in different fraction, and based on your their abundance measurement from the mass spec analysis. So one good example, so this data can be used whether the protein is eluting as a complex, because based on this SEC fractions, you can predict the size of that protein falling in that fraction. If the size of the protein is bigger than its monomeric mass, then you can decide whether, or you can tell whether the protein is eluting as a monomer or as a complex. If the protein A and protein B, they're physically interacting to each other, they should be migrating together when they're fractionated. So these two proteins, that information can be used whether these proteins are interacting with each other or not. So definitely it does not have the high resolution. So you might ended up identifying the hundreds of protein in its fraction. There are other challenges, and you can use other tools to confirm that. But this is also a very powerful approach when you couple this chromatographic based fractionation technique with your mass spec analysis. You can predict protein complexes and protein-protein interaction. So this is one of the example of the NDS dehydrogenase complex we identified in cyanobacteria, which are the photosynthetic microbes. And these NDS, the cyanobacteria, if they are photosynthetic microbes, they have the NDS has the two functions, one in the respiration as well as in the photosynthesis. So what we identify here, what we use this technique, we can identify the both forms of these NDS complexes. For example, in this CO2 uptake one, we are identifying some of these subunits which were missing in this respiratory form. So this allows to predict the function, yes, of the same complex or the same protein, but they have the different function. So whether it's becoming the respiratory complex, or is the CO2 fixation complex or CO2 uptake complex, can be gleaned for this type of information, which is really powerful when we want to interpret those data in the biological context. So far, what I described here is like the proteomics can give a lot of information if we could design the experiment in the right way. So just like here, what we can find is it gives you the quantitative information or expression level information. And then, so this will come in my later part of the talk, but if we design the experiment to do some kind of the targeted analysis, like the PTM analysis, phosphorylation, or glycosylation, or IP-based analysis, we can also modify our method and we can get that information. Like here, we are purifying this affinity, purifying the phosphorylated protein using this antibody or this affinity reagents. And the other one, we can also do this complex profiling using different these upstream chromatographic separation before we analyze them through the mass spec. So yeah, so that tells you the sample preparation is the key for a successful proteomic experiment. And then, one of the area I'm focusing recently is the cellular senescence, and I'm using this proteomics tool in combination with other molecular biology tools to determine the senescence. To investigate the senescence biology. And the experiment I started with oncogene-induced senescence, and I would like to go some of my preliminary data, and that will give you the much broader view how this proteomics data can be used in the context of biology. So we all know, definitely this audience knows well about this senescence. It's kind of this is a permanent cell cycle arrest. And this is the way the cells have evolved to prevent them from becoming tumorigenic or prevent from cancer. And there are so many different factors, so many different stressors that can induce senescence, like oncogene-induced senescence, the way oncogenes can be activated and the cells can enter senescence. And telomere shortening is one of the most widely reported one, telomere shortening, DNA damage, and other stressors. So one, as I mentioned before, the senescence is linked with the cancer biology. Yes, it's one of the mechanisms for the cell to protect from cancer. But when the senescent cells get accumulated, so it also becomes a problem. It also becomes it can also convert that anti-tumorigenic cell into the tumorigenic cell. And the senescence is one of the way that we become older. Yes, it's directly links with the aging, and that comes with the different age-related diseases. So understanding the biology of senescence and also identifying what are the molecular factors or the pathways that balance this senescence versus tumorigenesis will be critical critical for cancer prevention, as well as developing the new drugs or therapeutic targets for treating cancer and other pathologies like the neurological diseases. So there are two ways. I'll just go to a little bit faster of that. There are two different type of senescence there. So one is the DNA damage response dependent and DNA damage response independent. So different in this pathway, you can see this a P53, which activate these kinases. They activate the P53 gene here, which functions as a transcription factor and can activate this CDK inhibitor P21, and the cells enter into the senescence. And there is another oncogene P16, which is the inhibitor of this another cyclin-dependent kinases, and then the inhibition induces the senescence. And in the DDR, the DNA damage response independent one, there's a BRAF gene which activates this PPD. This is the PDP. This is the phosphatase which can dephosphorylase a PDK1 and causes the cells to go into senescence. So with the senescence, the cells release these pro-inflammatory factors. Yes, a SASP. So accumulation of SASP, the cells itself might be senescence, but it is this accumulation of the senescence and constant release of these factors influences the other neighboring cell. So even if that cell is senescence, the other cell can become tumorigenic. So there is a constant accumulation of those cells, is also bad. So there is a balance between the senescence and the tumorigenesis. So I mentioned before that the telomere shortening, because the telomeres don't have any protein, do not encode any protein. That's why in FC replication, they become short, and at one point, the cells has to enter into senescence. But in the meantime, if they can express telomerase, then the cells can replicate many times, then that becomes a problem, and the cell can become tumorigenic because of the uncontrolled growth. So this graph shows how the cell becomes senescence with the number of replication, but due to some mutation here, if the telomerase activity happens, then cells can go in this stage rather than this the senescence stage. So it's good to understand the biology of the telomere biology and how we can implement that for understanding the cancer biology. So based on that, so what I'm trying to do is there is a very unique structure in the nucleus, which we call the promyelocytic leukemia nuclear bodies, PML nuclear bodies. And then the PML nuclear bodies are the areas, the hub where many proteins they accumulate, and they can be targeted for degradation, as well as they can be activated when they accumulate in that sub-nuclear structures. So our goal here, the focus here is how these PML nuclear body proteins are regulated. PML itself is the major master regulator for the stability and function of PML nuclear bodies. So to study this, we are using these the IMR90 cells, and we have developed this ERAS induced the stable cell line, where the ERAS can be activated by treating them with this hydroxytamoxifen. So when you treat them, the ERAS is activated, and through ERAS activation, the cells go into the senescence. And we use this proteomics techniques, just I mentioned before, and we perform the global nuclear proteome and the phosphoproteome, and use this data in the context of the oncogene-induced senescence condition. So the first step is to look the phenotypes, and you can clearly see in the senescent cell, you can see that this green deposition, which shows the expression of beta-galactosidase compared to the methanol control. That says our cell, the induction and activation of ERAS is inducing senescence in our cells. Then we perform this phosphoproteome, and these are the numbers. I don't want to go to these numbers, and you can see we have captured the large number of phosphopeptides. But if you look at their temporal data here, this is the 0 day, and this is the day 2, day 4, and day 6. And this data, the phosphopeptides are clearly clustered separately there. That says there is a very nice response, time-dependent response to this senescence condition. So then we profile them, and this heat map here, this is the number of protein and phosphoprotein and phosphosites we identify. And if you come, we have about 20,000 phosphosites, which were mapped to 4,000 protein. So what does that mean is for each protein, we are identifying multiple phosphosites. So this heat map here shows these specific phosphosites. This is coming from these 20,000 phosphosites we map. So it's not only the protein level information. So what I'm trying to show here is that we are not only identifying phosphoprotein, we are not only identifying phosphopeptides, but we are also identifying phosphosites and their quantitative information. For example, if there is a protein A, which has 3 phosphosites, we identify they are phosphorylated due to the the OIS condition, in senescence condition. We can also measure which of those 3 sites is activated. Yes, are all the 3 are activated, or only 1 is activated? We can measure the activation or the abundance of each site they are phosphorylated. And also this is, I'm showing here another graph here, which comes from the liver sample, high fat diet mice sample. This is the senescent cell, and this is the liver cell when under a high fat diet condition. You can clearly see the differences. Yes. So this is a 0 day where everything, the phosphorylation is down, there is no phosphorylation. And when they enter into senescence, phosphorylation is significantly up. But the opposite is true in this high fat diets. From the low fat, when they enter into high fat, phosphorylation is down. So you can see, depending on these tissue types or cell types, or the treatment condition or experimental condition, the response of this phosphoproteome is opposite. So it's a very different. So I mentioned before the PML, proteolytic leukemia protein. This PML protein is the major protein in PML nuclear bodies, so major organizer. So it's very widely studied. And then so we're interested, what is the phosphorylation dynamics of PML when these IMR90 cells are going into senescence? So here we identified these 8 different significantly regulated phosphosites in this protein. So for example, I will be focusing more about this S527, and there is S17. These are different phosphosites we identified in the same PML protein. So now we have all these phosphosites information here, and all the phosphopeptide information, what we can do using this data. So one thing we first can look is, what are the kinases that are activated? We identified they're activated under senescence conditions. So this is what I'm showing here. For example, so this line here, y-axis, here shows all the protein with their particular phosphorylated site. For example, if you look this one, this is the PML phosphorylation site 575, and there is a PML phosphorylation 527. And this x-axis here, it shows the different kinase groups. So if you look the PML S75 and 527, you can clearly see there are different MAP kinases that are targeting these different sites. So this is MAPK3, appears to be very highly activated and it's phosphorylating S527, while this for 575, other kinases, there is this MAP kinase is also activated, but there are other kinases as well. They are targeting those. So you can see, you can not only identify, as I emphasized before, you can also precisely find which site is phosphorylated, as well as which site is mostly activated. And then you can also find the target specific kinases that are targeting those sites. And I think that information is going to be very, very powerful, very, very informative when we are linking them to the biology. So that's why, so we can do the network mapping after that. There are lots of public database you can find, and you can map this data with the publicly available data to see all this kinase, for example, MAP kinase signaling and other phosphorylation events. For example, this circle I'm showing here, these are the different MAP kinases we identified. And also the data, we can also link which MAP kinase phosphorylates to which substrate. Yes, this is, for example, MAP2K1, phosphorylase MAPK1. So MAPK1 is itself a substrate for this MAP2K1. So these black arrows here, I don't know whether you can see in your you can see or not, but the black arrows shows the direction of the phosphorylation, the events going on. So this way we can map not only MAP kinases and which direction the signaling cascade going on or the phosphorylation direction is happening. This is the graph. If you remember, this DNA damage response independent pathway I mentioned before. So we also identified this BRAF protein here, and this the MYC protein and BRAF protein, and this protein is known to be linked with this DDR independent the senescence process. And we know which sites of this is activated and what are the kinases that can go and target those sites. So this is, I believe this is a very useful information when we do the biology. So now, what other things we can do? So I'm running a little bit late, but I'll try to be faster here. So what we have all the information about the phosphorylated proteins. We have all the information about the kinases, all the sites that are phosphorylated, and also in most of the cases, all the kinase which are targeting those sites. Now, so what other information we can glean? So, it's where I believe you all know that the PTMs, the post-translational modification, are also interdependent. So the phosphorylation sometimes can derive the simulation and other modification, or it can also inhibit those processes. So can we find, for example, the simulation, because the PML, the protein, is known to be regulated by different simulation events. So, and in our data, we identified these two, PS1 and the RANB2. These are the E3 the sumo ligases, and clearly this protein, they appear to interact with the PML if we link those data. So what we're interested is, like, where in our next studies are those, how these proteins are involved in the senescence process. So what we can do in that situation, does the particular PML phosphorylation activity affects the simulation events or not? So we can go and target those phosphorylated sites. We can do some point mutation, or we can do the knockout studies and see what happens after that, what type of phenotype we see. So this is my next the study. What I'm interested is doing the PTM crosstalk and link the different phosphorylation sites with the simulation and ubiquitination site. And looking at simulation and ubiquitination is also interesting because they modify, they target the same, in some cases, they target the same lysine residue. So is there a competition between ubiquitination and simulation, and how that affects the downstream degradation is also very important or very informative. So that's why what we looked here is we also looked, these are all the proteasome complex subunits we identified in our global nuclear proteome and our phosphoproteome. And these are all these the proteasome subunits are there phosphorylated. There are other subunits that are also upregulated in the senescent cells compared to the control or the non-senescence. And this is the network here. Now you can see a lot of these proteasome subunits are phosphorylated. So the next question will be how this proteasome is involved in this process, yes, in the stability of the PML nuclear bodies, their function during the senescence. So what we can do? So we can go and now there are different very well-established inhibitors. Now we can go and inhibit those proteasome activities, or also there are activators, yes, stimulators. You can further stimulate those proteasomes activity and see what happens to the cell. Can you see the phenotypes? And we can do the same kind of analysis to see whether there is a degradation is happening, it is a proteasome dependent degradation, or it's a proteasome independent degradation. So this kind of information we can also glean for this type of analysis. So I want to spend a few minutes here. So because this what I'm trying to show here is the PML protein, and if you can see here, it's a modified in so many different sites. So I'm highlighting these two areas where we found phosphorylation of PML in this region. And we also found phosphorylation of PML in this region. And also PML is known to be sumolated in this region here. These are blue or green circles you can see. These are the sumolated lysine residues. So if you look the description here, you can clearly see when a certain site is phosphorylated, it has a certain function. For example, the phosphorylation of S8 by HEAP-K2 appears to link with the PML accumulation and also sumolation and apoptosis. On the other hand, so there are other areas like here, if you do the CDK12, then which we also found this 527 and 518 phosphorylation, and it related to PML degradation. So the apoptosis here is driving the cell towards more tumorigenic conditions where this is a PML degradation. But the interesting part is in our total PML, total proteome, global proteome, PML level was not changing. It was the same. It was not significantly different. So that raises the questions, is there a PML degradation is also compensated by PML synthesis? Yes, there is, or PML recruitment to the nuclear bodies. So what is the proteostasis condition there? So we can measure we can get those kind of information by these studies. And you can clearly see one thing I want to highlight here is this site-specific phosphorylation is clearly linked with other PTMs like a sumolation, ubiquitination, and their proteasome-dependent degradation. So this phosphorylation site-specific information is really powerful and really insightful to link this the PML nuclear body biology. So the next step, what we are trying to do is we are going to perform the similar studies in sumolation and ubiquitination, and also we find this PIN1, the isomerase 1, which is known to which isomerizes the PML, and once it gets phosphorylated, and it has this a very specific phosphorylation sites. The PML, if the PML phosphorylates in that particular site, then only PIN1 can function and can isomerize it. So isomerization of PML can have the other downstream effect. It might help it to go sumolation and then ubiquitination and degradation, or it might also inhibit sumolation because now it's isomerizes. That means the sites might be less accessible for other types of modification. So we have generated this very nice PIN1 knockdown lines in the IMR90 cells, and we're going to explore how this knockdown affects other PTMs and the cell phenotype. So the idea is we can go and do the different specific knockdown or inhibition of these kinases, or the mutation of the particular modified site, or knockdown or knockout of the gene, because we have some certain target, these E3 ubiquitin ligases, we have certain highly activated kinases, so we can go and inhibit them or knock down some of the genes and look how that affects the PML nuclear body stability and function, and how that stability and function can be linked to senescence. So this is my last slide, and I hope I was able to convince you this proteomics is simply not a technique for generating the list of protein. It's a very powerful biological tool, and it can be used not only test the hypothesis, it can be used to generate a new hypothesis or discover new biology. And it's very, as I showed, that only the 27% story can be linked to RNAs, so 40% can be linked to the protein. So it nicely complements in other omics approaches. And it's a very good, powerful tool to identify global targets, just like we identified the BRAF and the different E3 ubiquitin ligases, different kinases there. These are the global targets which can be used later on for mechanistic studies and phenotypic studies using other molecular biology tools. And site-specific PTMs, I believe I convinced you that site-specific this modification information are really biologically insightful, and they can derive a new biology and a new ideas in the field. One area definitely going to be very critical is the kinases. So we are going to look some of those kinases we identify in our future studies. And as I mentioned, the crosstalk between stimulation, phosphorylation, and ubiquitination is going to be focused in my future studies. So with this, I would like to stop here and acknowledge the contribution made by different people in my group. And Rodrigo Mohalem. Rodrigo is my graduate student, and majority of the data I showed you today was his work. So he joined my lab in 2020, and he's a graduate student, PhD student in the Department of Comparative Pathobiology. And Dr. Jacqueline Franco, she is a lab manager at the Proteomics Facility. She's an expert in not only proteomics, she's also expert in the lipidomics and the small molecule world. And she comes to us from Purdue too, and she was the graduate student in the same department I belong to, comparative pathobiology. And Jacqueline started in 2020. Then the Sora Kim, she was our graduate research assistant in my lab in 2020. And the high-fat diet, the heat map I showed you to compare how the phosphoproteome profile differs between different tissues or different conditions, that work, the high-fat diet, liver proteome, and phosphoproteome work was, she contributed to that work. Then Susan is our building manager, and she helps us in day-to-day operation. She's a amazing resource for us, and anything we need, she's the first stop. So I would like to acknowledge her contribution to run the core facility efficiently, as well as to make for my independent research. And Dr. Wen Wang. So Dr. Wang is the director of the Purdue gene editing facility, and we work very closely with him, and he's our great resource when it comes to gene editing and other mutational studies. And I also like to acknowledge the collaboration with the Trader Lab, Andersoni Lab, and the Bowman Lab at Purdue, and the funding from Indiana CTSI, so Walter Trust, and the Purdue EBPRP. With this, I will stop here, and happy to answer any questions you may have. Thank you.
Thank you, Uma. That was really very informative. Particularly, I really like the way you showed the application of these analyses to your own studies. We do have a couple questions in the chat, so we'll go ahead and start there. Andy Yu asks, for the protein sample preparation, how do we prepare detergent-resistant proteins, such as membrane proteins or lipid-enriched proteins?
Great. This is a great question, and this is the challenge too we face each and every day. So there are a couple of things we can do. Yes, one is if you need to use the detergents, and I forgot to mention, detergents kills our system. Yes, they're really, really bad, and they're very sticky when they go to our LC column, and they protonate so easily. And then all the signal we got from the detergent, so it masks the signal from our peptide, which are difficult to protonate. For the membrane protein, so we typically do two approaches. One is if you really need to do the detergent, like you need to use SDS. We separate those protein after you extract them in SDS, a buffer, we run through the gel. And in the gel too, we decide to run two types of gel. One is we call the quick gel. So when you extract the protein, you then pass through the gel and run a few minutes, 5 or 6 minutes, which we term the quick gel. And then you cut that, the gel area, and we do the in-gel digestion. That's the one way to do it. Once you run through the gel, the problem of the SDS or any other detergents can be resolved to large extent. So that's the one thing. The other thing also we found it might be useful is when you extract the protein, for example, from the cell and tissue, you take everything. Yes, if they use the buffer that is compatible with digestion, like that does not inhibit the activity of trypsin and lysine. So you lyse them and you take everything, whatever you need, and you start digesting them with all the debris. So what happens is when the digestion starts, so if there's let's say the transmembrane domain, they start chopping out this hydrophilic domain side, yes, and then they start cutting those peptides out, the proteins out. Then you move on, the solubility, the extraction of the remaining part also becomes more easier, and this the protein become more and more accessible. So we found that useful. In that situation, you might need to sometimes do the multi-step process, rather than doing, like say, 3-hour digestion. Maybe you do the 3-hour and add a little bit more trypsin or other proteases and let it go for 2 more hours. Or we also sometimes implement the high pressure digestion system. So rather than doing in the room temperature at 37, we go and digest them at like 30,000 PSI. These are use the barocycler and cell disruptor, barocycler cell disruptor, and we digest there. That also helps. So there is no one strategy fits all situation, but this is what we do here. And the other, maybe I may add, other thing is you can also do the soluble and insoluble prep. Yes, just like you lyse them and you first extract all the soluble protein, which are not difficult to extract. Then you got the pellet and digest the pellet separately. And then the soluble portion, soluble fraction separately. So instead of having one sample, now you can have 2 samples. But this also, we found that significantly help us to identify more this membrane protein and also increase the depth of our proteome coverage. I hope this answers Andy's question.
So the next question is, are there general enrichment methods for most PTMs? For example, if I wanted to look at global methylation on proteins, is there a preferred way of enriching methylated proteins or peptides?
They are not. Yes, that's the challenge. The one area that's a very well established is the phosphorylation. So when it comes to other like glycosylation and phosphorylation is pretty well established methods there. When it comes to other like the sumo and ubiquitination I talked, there are not easy way to do that. So people use the antibody-based purification and so many different other strategies. So same goes to the methylation too. So the one way to look at it is if there are specific antibodies and we can do the IP or pull down based on the antibodies that people use. And the other ways is the people, if the protein is if known to be located in a certain organelle, people also go sometime purify this organelle. Like if it's a nuclear protein, for example, yes, we can go and purify the nuclear fraction and use that nuclear fraction and with these antibodies to pull down the protein of target before you characterize. But unfortunately, they're not well-established regular methods that we can do large number of those PTMs. That's a huge open area, and looks like it's not as easy as doing the phosphorylation or glycosylation studies.
Okay, and then another question is, this is actually kind of a 3-part question. I'll just give them to you one at a time. Could you comment on the comparison of MS-based proteomics and RPPA profiling? So maybe define RPPA for people that might not know.
Okay, RPPA.
Yes. If you don't know what it is, maybe
what is the RPPA?
Maybe you could
yeah. Can you
reverse phase protein array?
Oh, okay. So it's a reverse phase protein chromatography. Okay, sorry. So it's like that's a two. The MS is the MS part. Yes, is that the reverse phase? I don't know. I want to make sure that's what
reverse phase protein array.
Okay, it's I'm not much knowledgeable in the protein arrays world. Yes, so I can explain a little bit about the reverse phase. That's pretty much the LC, the part we use. It's more like the reverse phase chromatography system, but the protein arrays world, now that's not an area what I will like to make a comment where I'm not well aware of. Yeah, so I know this term, but I don't have the expertise on that area to comment and compare these two systems.
Okay, and then does the facility also provide service on metabolomics?
No, we have our next door, there is a metabolomics core. The Bruce Cooper, Dr. Cooper is the director of metabolic profiling facility. So we have the separate dedicated core facility for metabolomics and also lipidomics.
Eric, you can go ahead and ask. You can unmute yourself.
Well, no, I saw that that person had one more question, so you can finish asking and then I will go.
Okay, the other question is, for folks outside of Purdue at Notre Dame or IU, do you offer internal pricing or external pricing for your services?
Yeah, very good question. And I struggle every time when I get a request there. So I think the strategy has been it's a confusing. But if we are the CTSI core, if it's the CTSI core, my decision is we should provide the internal rate, and I want to stick with that. But there are some other like some sort of contradiction there. But I definitely whether this is the CTSI funded project or not, but I try to make it internal. And I think as a director, I can do that. So I haven't got any complaints so far. But this is the debate we sometimes face. But the understanding is, for me, for PPF, I can speak only PPF. Yeah, if it's Notre Dame and IU and Purdue, we stick to, I want to stick to the internal rate.
That's great. I'm sure everyone's happy to hear that. Yeah. Go ahead, Eric.
Yes, I have two questions. The first one is regarding a protein-protein interaction identification. I understood that the exclusion site, size exclusion chromatography is the key step in that analytical workflow. But then if you have in fraction number 2, for example, yes, your protein interaction or the target, but also you have other proteins. How can you recognize the two proteins that are interacting?
That's a very good question. That's why if you recall, I say the resolution of the chromatography system is not great. So you ended up identifying hundreds of proteins. Yes. So, and how do you do that? It's a challenge, but you have to add other steps on that too. For example, if you're really doing the high throughput or high level protein-protein interactome, what you can do is you can go and use the second step separation with the ion exchange chromatography system. So like you have this size-based fractionation, then take that fraction and you go and separate them by another chromatographic system like the ion-based chromatography system. Yes. So then, because this is a completely two different separation, then can you see these two proteins now? Now you are with every step of separation, you are increasing the resolution, and ion exchange columns are much better, has a higher resolution than the SEC column. So then if you see that two protein A and B are there, yes, still they're migrating together, you can say they're physically interacting. That's the one way, but that's not the only. So the other thing we try to do is like use different computational tools. So if the protein, even if you have a 500 protein in that fraction, yes, instead of having 5,000, now you have narrowed down at least 500. That's a 10-fold less. Now you can do this different, the clustering algorithms. And in the cluster nodes, you can define from that 500, how many clusters you want to generate. Okay. Let's say you generated 20 clusters, correct? In that 20 clusters, now you divide this protein, some cluster might have bigger, some cluster might be smaller, but where that protein A and B fits. So do they cluster nicely together? For example, the proteasome complex I showed you, if you cluster the whole proteome, all the proteasome subunits will cluster together. Yes. And there are very nice tools out there where you can go and does my protein A and B are falling with that one within the same cluster or not. So that's the computational way to look at too. Yes. And then does that make sense?
Yes.
Yeah, and then the other step, which I found really useful, I didn't show the data here, but when you do the IP experiment, for example, yes, so there was a situation when I do the IP to validate some of my proteomics data. So what I found is there is a protein called the nitrilase, for example. So in the plants, so the nitrilase, they have the 4 different isoforms. And when you do the IP and with the nitrilase antibody, you are pulling all the isoforms, but also I'm always pulling this thioredoxin. And even if no matter how much I clean my sample, how much I do, I also pulling down thioredoxin. It behaves exactly the same way. So if you don't have another information that I'm coming a little bit in a few seconds, so you will think that thioredoxin interacts with nitrilase if you only look the IP and mass spec data. But the nitrilase is cytosolic. Thioredoxin is a chloroplast protein. Okay, there is no way I can, because I know that, yes, then no way why the thioredoxin will be with my IP. So if you don't have that information, or you are a little bit naive about the mass spec, then what happens is people go, now the thyroidoxin is interacting with nitrulase. But then what I did is, now I went and looked my SEC fractions. Yes, does nitrulase and thyroidoxin migrate together? The answer was no. There was no trace of thyroidoxin where the nitrulase is peaking. So nitrulase is coming way in a different fraction than where the thyroidoxin are peaking. So that when I combined that with my SEC data, it helped me to say, no, this thyroidoxin is a chance pull down. Yes, it's a contaminant there. So what is happening is it's so abundant. Thyroidoxin is so abundant. Now you are pulling down from that 5,000 protein, only few protein in your IP. Now, even if you have a 0.001% of thyroidoxin to pull down in your sample, that chance pulling, it's still enough to ruin all your experiment. Yes. So there are certain advantages if you really critically think and implement those tools. But I agree with your comments that SEC or IX alone cannot find all the protein, whether the protein A and protein B are really interacting each other or not, or what are the new interactors. If you want to find, we need to integrate those different chromatographic system with the computational tool and other like the IP and other type of steps.
Sure. Thank you very much, Professor. This is not my second question, but it occurs to me after listening to you is, I guess the advantage of proteomics in comparison with native SIMS is that in native SIMS, you need to take care of the solubility, of course, of the complex of protein, while in proteomics, you forget that part, right?
Yeah. Yes,
okay.
True. That's and in the SEC profiling too. Like when you do the SEC, the goal is you are not denaturing the protein. Yes, so that's you want to use the protein as native as possible.
Yes.
Then the question, that's why at one point, if you remember, I made there. So the protein, they're very concentrated. It's like they're working inside the cell as a soup. Yes, there is a 300 milligram protein per milliliter. So you can imagine that's a 2 or 3-fold, 200 times or 300 times more concentrated than our typical reaction we do in the biochemistry lab. So when we're extracting, we should also consider those things. You put let's say you're a small tissue, you're putting 10 ml of the buffer, so with the hope that you can by using more buffer, you can solubilize more. That's not the case, because when you dilute them, so it's significantly dilute than they were supposed to be. Yes, then you are already destroying some of those interactions there. Protein cannot interact in a completely different concentration condition. They have to have some specific conditions to interact each other. So when you apply the buffer, the volume of the buffer, size, balancing the size of the tissues versus the volume of the buffer, you should be thinking those two. They don't. If you dilute too much, you're already destroying. If you want to get that native information. If you don't care about that, only the identification and quantitation, that's fine. But if you are doing the protein-protein interaction, we should consider those things before we design our sample preparation stage. So that's a very critical and very useful.
Thank you, Professor. Do we have time still for my second question?
I actually have to kind of wrap things up because I'm going to leave, but maybe you can follow up with him by
Yes,
email.
please send me email. I forgot to put my email, but I'm at Purdue. Please feel free to send email and I'll be happy to answer questions.
Sure. Thanks very much, Professor. Very informative. Thank you.
We just have one really quick question. I think it'll be very fast answer is, the question is, I will use antibody protein A magnetic beads to pull down a membrane protein. I cannot use SDS in the lysis buffer. Which detergent do you recommend? Triton X100, NP40, or CHAPS?
None of those for mass spec. Yes, everything is bad for mass spec. So, but the one thing is, like if the question is only what you can use for solubilization. If there is no mass spec component, yeah, you can use those things. Yes. And there are also some nice, I forgot the name, they're more membrane solubilizing and non-ionic detergents too. So if it depends on whether that's compatible with it or not. But if it's a mass spec thing, yes, if the mass spec thing, and you can use SDS, if the SDS helps, yes. But as I said, then we can do like a gel-based separation. You pull down with your beads, And we can run that through the gel, and we can analyze by the mass spec. If that's the case, if the SDS is coming on the context of mass spec, yes, you can use SDS, you can pull down, and that IP now we'll run through the gel, quick gel, and we run into the mass spec. But if the SDS is not compatible with your pull down, the NP4D looks like the most widely used, yes, is a very common, but there are also very nice this membrane solubilizing other reagents too. I think the DDM, so that's the one people use sometimes to solubilize the membrane, and they say the DDM is more compatible with the mass spec analysis too. But I don't have the experience of running those samples in our system. I hope that answers, but again, if I'm just answering the oft. So please let me know via email, and I'm happy to discuss it.
Well, great. This was a really great Q&A session and a great seminar. And thank you everyone for coming. And please go, if you're interested, you can go to the ATB site and look at the video recordings. So thank you, everyone.
Yeah, thank you so much. Thanks for your time.